Good afternoon, everybody. Buenas tardes a todos. Es un gran placer presentar a Jerry Carter. Hello, everybody. Welcome to the last talk of today. And it is my great pleasure to be introducing Jerry Carter, who's going to talk about the social lives of vampire bats that you're seeing in this video right here. So I'll start with a very brief background. Jerry did his undergraduate at Cornell University, then a master's at the University of Western Ontario, and a PhD at the University of Maryland with Jerry Wilkinson. I then had the great pleasure of hosting Jerry for a postdoc in my lab at the Smithsonian Tropical Research Institute in Panama, which he followed with a postdoc at the Max Planck Institute for Animal Behavior in Constance, Germany. And if you're looking closely, Jerry, these pictures don't quite match the eras, but I worked on it. And then in fall 2018, Jerry began his current position as a professor at The Ohio State University, where he currently leads a vibrant and extremely productive lab group. When I recently asked Jerry for some in-action photos of him in the field, he told me that most of his in-action photos these days look like this. I know this is not entirely the truth because I saw him catching vampire bats in Panama just a few months ago. But it's been a huge delight to see this little Jay arrive on the scene, and also it's been really inspiring to see how Jerry and Michelle are balancing family and science. So last year, with overwhelmingly strong support from his students and colleagues who nominated him, Jerry was presented with a 2021 ABS Outstanding New Investigator Award. And just before I conclude and pass the floor to Jerry, I want to read this note sent to me by Mae Dixon, who's now a postdoc in Jerry's lab, because it captures Jerry's character perfectly. I have to take my glasses. So it makes complete sense that Jerry studies friendship and cooperation, because Jerry is remarkably pro-social and is the best cooperator I've ever met. Anyone who's collaborated with Jerry knows that he routinely does far more than his share of the work. He manages this despite routinely getting sidetracked into hours-long conversations on anything at all. He's such a good scientific role model. He is dedicated to transparency. He actively tries to falsify his own hypotheses, and he's the first to admit when he's proven wrong. Jerry tries to live by his belief that compassion and rationality should guide all of our decision-making. The vibrancy and diversity of his lab is testament to this strategy. And with that, I'll turn it to you. Welcome, Jerry.
Oh no, this is like the worst way to start. Oh, just give me a second. Oh my goodness. Thanks so much, Rachel. That was really nice. That was super nice. Okay. Okay, I'm just going to jump right in. If I start saying anything, I'll go off on some long tangent. Okay. I want to start with this quote I really love from James Clerk Maxwell, of Maxwell's equations. Thoroughly conscious ignorance is the prelude to every real advance in science. I really love this quote. And this is a talk about vampire bat cooperation, but I think the most important thing I can say is that I don't really understand the social lives of vampire bats. That's the thing I try to remind myself of every day. And I don't know how many hundreds of hours I've spent watching vampire bats. I've done a bunch of experiments with them that I'll show you. But their social life is still fundamentally mysterious to me. So I really want to be explicit about that. And so what I'm going to do is every time I draw a conclusion, I'm going to put this little confidence label down in the corner. Low means I think this is true, but I wouldn't be shocked if somebody shows that this is wrong in the future. It means this is the best evidence suggests this is true. Medium means I would be really surprised if this wasn't true. It means that there are multiple lines of evidence for this. And high means this is a core assumption that I don't really question anymore. I treat this like a fact. So I'll say that I first became interested in this topic of of cooperation, sociality, and vampire bats as an undergraduate. When I read the book, The Selfish Gene, the second edition features the PhD work of Jerry Wilkinson, which was done here in Costa Rica at Santa Rosa and La Pacifica. It was published in Nature in 1984. It's entitled Reciprocal Food Sharing in Vampire Bats. I became really obsessed with this study. I remember seeing this photo in the popular article in Scientific American of a person's legs sticking out of a hollow tree, which is what you're seeing here. That's a lawn chair, and this is because they were spending hundreds of hours watching the bats inside these trees. And if you haven't had the pleasure of going inside of one of these roosts of vampire bats, it's like crawling into a latrine, like going into an outhouse through the wrong door. So the amount of guano and urine that must have been raining down on them, it's just yikes. But I remember thinking, wow, that's dedication. I also remember thinking, I could do that. I could crawl into some trees. But I was really impressed by what they found. The basic story of this paper is that vampire bats just get this one blood meal a night. If they miss 3 nights without feeding, they'll die. But they have this social safety net. They can come back to the roost and be fed by other bats. So females feed their offspring with regurgitated blood, but they also feed adults, including unrelated adults. And the food sharing is very kin biased, so it's 95% of the donations that Wilkinson saw were between close kin. But then Wilkinson took away the relatives, and he had these groups of completely unrelated adults, and they fed each other in this reciprocal fashion. And it was a pretty small sample size, sorry, Jerry, but the pattern was really striking. And he concluded that food sharing was stabilized by both kin selection and reciprocity. This was back in 1984, just 3 years after Axelrod and Hamilton had published their famous paper modeling reciprocity as tit for tat in the iterated prisoner's dilemma. And over the next 3 decades, this became this textbook example of reciprocity, but reciprocity itself also became increasingly controversial. So in 2010, when I was just starting my PhD work, Tim Cluttenbrock had published a review paper in Nature saying there was really no evidence of reciprocity outside humans. And so this pendulum of the importance of reciprocity has kind of swung back and forth. The main alternative explanation is what Richard Connor called pseudo-reciprocity. Basically, for this case, it means that food donations are this costly cooperative investment that the bats make, but the returns on that investment are a byproduct of the selfish or self-serving behaviors of the other bats in the colony. So there's no real problem with cheating. So group augmentation would be an example of this kind of byproduct returns. So I've been studying this now for a few years, and people still come to me and say, hey, is food sharing in vampire bats actually reciprocity? Should I be teaching this in my animal behavior class or not? Like, what's the current state? And every year I say, I'm about to resolve this. So next year I will tell you for sure what it is. But to really answer this question, I need to explain how I've come to see the evolutionary explanations of cooperation. So when I started my PhD, the literature on this topic was full of these categorical trees. So each path in this tree is a different explanation for cooperation. This one is by Stuart West's group at Oxford. It's probably the most influential, I would guess. I don't know. But there's constant source of debate and disagreement about these because there's actually a whole set of these semantic frameworks, and they're all slightly different. This is just a sample. So these were all published around the same time. I remember 2006, 2007, all these papers came out, slightly different definitions of terms. So when you talk to people from other labs, there's this endless disagreement because different authors, different labs have slightly different definitions of terms like reciprocity or cooperation. I'm sorry to say this is really still the state of the field 15 years later. So if you're a graduate student getting started in this field, you have to just learn what everybody means by these different terms if you hope to really communicate with anybody. And I would say that in general, people tend to agree with these 3 basic factors being important. So you have indirect fitness benefits, kin selection on the right. Then on the left, you have cases where, like I said, cooperative investments provide byproduct returns or pseudo-reciprocity. But I prefer to use Gilbert Roberts' concept of fitness interdependence because that's a continuous variable rather than a category, and I'll explain why I think that's important. Then we have this conditional enforcement category. Basically means you're more likely to cooperate if your partner cooperates with you. So this could be partner choice or partner control, like you're rewarding or punishing based on what your partner does. Some people hate calling this enforcement, and some people call this reciprocity, but no matter what you call it, somebody's going to not like what you're calling it, so you can't really win. So I'm going to actually use this other term from the interacting phenotypes literature, responsiveness. I think that's a good term because it's a continuous variable, and also it's a really conservative term that's really stating what it is we're actually measuring. We don't know if it's having such an enforcement on cooperation, but we know if an animal's being responsive or not to its partner. So these categories are really useful, but a common problem that I see when I review papers and when I see talks, I think, is that people can get confused into thinking that these are mutually exclusive alternative hypotheses, and they're really not. They're actually continuous factors that could all coexist and interact within the same system. And I think having these categories and these models of extreme cases can lead to these sort of false dichotomies. So just as a concrete example, why are these chimpanzees grooming each other? Is it reciprocity or is it pseudo-reciprocity? Is it a social bond where they have fitness interdependence, or is it an exchange of a commodity driven by like a biological market of partner options where there's no partner fidelity? And I think we don't really have to choose between these categories if we think of these things as continuous factors. So rather than asking, is it reciprocity, we could ask, how much responsiveness is there? How much interdependence is there? How much partner fidelity is there? It becomes more of a measurement problem rather than a sort of like test this hypothesis, or is it this one or this one? And I think that maybe this is already obvious to some of you, but we already know that cooperative traits can be stabilized by multiple factors, and we know this from the best studied systems, so eusocial insects and bacteria, Dictyostelium, the slime mold. In all these cases, you see there's multiple forces that stabilize cooperation. And I think this is probably often the case, not just sometimes the case. And even if we take eusocial insects like ants as an example, and I'll use ants because we really understand ants way better than vampire bats for sure, but also I would say primates as well. We understand cooperation ants much better. And even there, with this very extreme case of altruism, the most extreme kind of altruism, we see there's really multiple factors involved. So we know relatedness leads to indirect fitness benefits. Eusociality only involved in lineages that were monogamous families. So kin selection is obviously pivotal here. But there's also this between-group competition, which creates interdependence between ants within the same colony because they're going to live or die together. And you also have direct fitness interdependence, for instance, when you have multiple queens that come together to found a colony. And we also know that despite all of the alignment of interests here, workers still, in many cases, selfishly reproduce, and then this cheating is suppressed by worker policing, which is a form of enforcement. So there you have enforced altruism. And this, again, is responsiveness to cheating. And so these explanations are not mutually exclusive. They're continuously variable factors, and you can think about them as being turned up or down across species, across scenarios, or in the same scenario over time. And I think what we should be thinking about is, what's the relative importance of these things, and how might they interact, and do they interact? Something else that we get from thinking about this is thinking about statistical power, right? So rather than thinking of reciprocity as a hypothesis, if it's something that could be a little bit or a lot, it means that we have to start thinking about what's the power needed to detect a small amount of responsiveness. Something else is thinking about possible interactions between these things, which not a lot of theorists have done, but there's increasing work on this. So Van Cleve and Akche had this theory paper talking about interactions between like responsiveness and interdependence, and as you turn the dials up, the game structure changes. And a prisoner's dilemma can become a snowdrift game, and as you turn them up together, these payoffs can change in these very complex, unpredictable ways. And it makes us think about how these factors can change over time, right? So if you ask the question of what's the game payoffs here, it could be that those game payoffs are fluidly changing, not only over evolutionary time, but during the organism's lifetime. I think a really good example of that is social bonding, right? So what changes when animals form social bonds? If we think about these explanations for cooperation, we might imagine that they have higher fitness interdependence, meaning that one individual starts to have more of a stake in the success of another individual, their survival of another individual, because that individual's survival, reproductive success feeds back on your fitness. And there might be a lower probability of partner switching, less of a threat of partner switching. And so maybe there's more or less regulation of the relationship. So we can imagine that these dials might be turning. And so much of what we're doing now is sort of thinking about these extreme cases. So tit for tat is the most extreme form of responsiveness. And the prisoner's dilemma is the most extreme conflict that you can have and still have cooperation. And if we go in the other direction, we say, well, they have a social bond. So their fitness is aligned. But that's assuming that there's no competition, there's no cheating, there's no conflict. And most of the cases are going to be in this gray middle area over here where I don't know if we even have a name for this. You could call it reciprocity, you could call it pseudo-reciprocity, depending on what you're using as your definition. But most cases are, I think, going to be in the middle. And this argument is made really beautifully in this paper by Redwin Bashiri, 2016, in Philosophical Transactions. The title is something like, why most cases of helping have intermediate levels of conflict. The other issue is that there's really a bewildering number of possible factors that could be important, and we have to just realize we don't really know. And these things are the kinds of things we care about as behaviorists. So why does one chimpanzee groom another? Spatial proximity could play a role, but also the market of other possible partners, and also the recent interactions and the long-term history of interactions and your kinship and the partner's dominance, and also your experience that you've had with other individuals. So in dominance, this would be winner-loser effects, and cooperation, this would be generalized reciprocity. That could feed into how you interact with this individual. You might have a sort of personality trait that makes you predispose you to be more cooperative in general. That has an effect. And individuals might differ in how responsive they are to different partners. And then the partner has these traits that could feed back on the actor. And so it's hard, it's a bit daunting to think about all of these different things and how if the effect sizes of these things are small, it'd be hard to detect them, and that doesn't mean that they're not important. And these effects probably are nonlinear, which again is just even worse, and they could all be interacting with each other. So I think we sort of ignore how complex these things might actually be. And they could be confounded with each other, meaning that you have to experimentally manipulate each one while holding the other constant. That's really hard. And perhaps the most difficult thing is when you're talking about cooperation in the context of a stable relationship, it's difficult to manipulate because social bonds are by definition stable. That's what they are. So the question to me is like, how well could we manipulate relationships using these factors? How well can we play matchmaker and sort of either predict or sort of change who's gonna be friends with who or who's gonna bond with who? And I don't think we know the answer to that question, but the more we can do this, the more we can claim to really have an understanding of how social relationships work. So that's what I'm trying to do with vampire bats in the long term. And I think one reason vampire bats are good for this is that they have a lot of behaviors that we can measure that are cooperative, and they're also just really small. And that's useful because all of the social drama of their lives play out in these very small spaces. It's like the size of a closet. And we can simulate that in the lab. So we bring them in the lab, and we can see that they do a lot of the same things in the wild that they do in the lab. We can induce cooperation as many times as we want. We can create new relationships. So here, introduce a stranger to some familiar bats. And sorry, I'm getting really sweaty in my... and we can track their social behavior in the wild. Is that too loud, or is... Okay. We can track their social behavior in the wild with these proximity sensors. These were developed by Simon Ripperger, who did a postdoc in my lab. He led a team of engineers to build these. They give us these really high-resolution social network data for, we can get them from every minute or hour or day up for about 10 days because we glue them on the fur and then they fall off after that. And using these, we can do things like we can track relationships from the lab to the field. So in the study, we captured wild bats, we brought them into the labs. We had this almost 2-year period where we had bats in the lab and we see their relationships forming, their grooming and food sharing increasing and strengthening, and then we could release those individuals back into the wild where they have hundreds or thousands of other individuals they can interact with, and we can see the stickiness of that relationship persisting in a different environment. So I should sort of come back now and talk about just vampire bat social structure in general. So what are the basics? What do we know? So most of this is mapped out by Gerald Wilkinson in the 80s. So males disperse at adulthood, and you get there's a female phyllopatriary, so you have multiple matrilines of females, but there's new females that join a group occasionally, which means that they're now unrelated to all of the bats in that group, and they don't know any of those bats, and they have to form new relationships. The kinship in these groups is fairly low, 0.08, and there's preferred associations that form between both kin and non-kin, and there are these fission-fusion dynamics, meaning the groups break apart and recombine. But these fission-fusion dynamics occur at multiple scales. It's really interesting to think about what these scales are. So the first level is that bats can choose to roost in the same hollow tree or not, or they can switch between trees. And we can make a social network at this level. So we can make a co-roosting network, and we can see the bats have preferred co-roosting partners, and that you can see that there are these emergent communities. But inside the tree, there's still fission-fusion dynamics going on because you get these clusters that form. The bats are moving between these clusters, and we can see this on video, or we can look at it with proximity sensors. And then within a cluster, bats that are sort of huddled all together, the bats are still making decisions about which of the bats around them they're going to direct their attention towards. They might spend more time grooming particular individuals or feeding individuals, and we can make a network. At this level, we can make these directed networks of grooming or food sharing. And what's interesting here is that each spatial scale constrains who can interact with who in the next scale down. And one thing to note from this is that all else being equal, bats that move around more at each scale are going to be more socially connected. I'll just leave it there for that, because if you want to hear more about this, you should just, well, let's talk about my graduate student, Raven Hartman. She made an agent-based model using real data of bat movements to try to disentangle the effect at all of these multiple scales and how they collapse down to the grooming network. So her talk is relative impacts of different spatial scales of movement on social grooming networks and vampire bats. It's Friday in the social competency session. But we also know that movement increases social connectedness because we've manipulated it experimentally. So if you inject bats with LPS, this challenges their immune system, they get sick, they move less, and this by itself reduces their social connectedness. This work was inspired by this really amazing work done by Patricia Lopez, who I haven't met. I'd love to meet you if I think you're here at this conference. But anyway, sorry. And yeah, so we did this. We just copied your work, sorry. We copied it. We did it with vampire bats in the lab and in the field. And this work was led by Basti Stockmeyer. He's now moved on to, we've got these sort of opportunistic data sets because we've had outbreaks of staph infection and rabies. We have real pathogens now and looking at how those shape the networks, but that's a whole nother story. Am I already going off topic? Okay, back to the talk. So movement rate is not a social trait, but these bats do have social traits that I think are variable between individuals. So for instance, when we introduce several, like a panel of strangers to several females, we find that some of those females are going to go and groom all of these bats, day after day, and others basically don't interact with those bats. It's like almost like a kind of introverted, extroverted thing. And also, the bats that are grooming more of those bats, these are all new bats they've never met. They also tend to be bats that are feeding, that are doing more food sharing later. And also, we see that bats are really they really vary and they're responsive to the same set of playbacks. So social structure is being shaped by variation at the individual level. There's also variation across relationships, and they have very differentiated social relationships, and we can see this across multiple different kinds of interactions, which all sort of correlate with each other. So clustering in the lab correlates with allogrooming in the lab, which correlates with food sharing in the lab, which correlates with clustering in the wild, and with social foraging in the wild, just with the same individuals across these contexts. And they're spending, most of their social interactions are happening inside of these very tight, completely dark spaces. So they're not roaming around a whole lot. And this plot is showing, sorry, I don't know, got scrambled a little bit there, but this plot is showing how much time a bat is spending inside the roost. So that colored bar is the bat outside the roost for one night. So that's the time outside the roost for one bat on one night. So each bat is a different color, and 6 p.m. is the blue line at the beginning, and then 6 a.m. sunrise is the blue line at the other end. And you can see the bats are basically spending all their time inside this completely dark tree, and they only emerge, they might only be outside for like an hour or two, and then they come back. And so what are they doing when they're inside that tree? They're awake. I mean, this is all awake time, but they're socializing, they're moving around, they're calling, they're grooming, they're sniffing each other. Who knows what they're doing? I know, that's what I'm trying to understand what they're doing in there. I don't know. So then they leave, and they forage on blood. They feed on nothing but blood. They'll feed on just about any large animal. Usually when we're studying them, we're feeding on livestock, but they'll feed on anything. This is an image of a bat feeding on a penguin. That one's feeding on a mountain lion or a puma. They have a really diverse set of hosts. And they do social foraging. So there's this mix of affiliation and conflict that we see during foraging. We see some bats will fight over a wound, other bats will feed peacefully from the same wound. It's an open question about whether there's producer-scrounger dynamics, because once a bat makes a bite, you've created a resource where other bats can come and drink from that wound. But what we have seen is that these affiliative interactions they're much longer in duration, and we see that those are the ones that are between bats that we know are affiliated within the roost as well. They make a whole bunch of calls when they're social foraging that we've linked to the behaviors because we've synchronized these ultrasonic recordings with video, but we don't really understand this very much. This is something we're trying to understand more. The one call that we do understand something about are these contact calls, because they make them in isolation when we pull them for these fasting trials that I'll tell you about in a second. So contact calls, we know that they recognize individuals from these calls, and they can actually literally call in their food sharing partners. So if you have a bat and you play a high donor that's unrelated and a low donor that's unrelated, they show a really strong, obvious bias towards the bats that have fed them before. And how do we know who their food sharing partners are? Well, we can induce food sharing. So when we fast a bat, as if it had failed to feed, and then we put it back in and we see who feeds it. And this is Jerry Wilkinson first did this back when he did his study in 1984, but he only did it a few times, and then I basically just done it way more times. So I've just done this, how many times have I done? Over 1,200 times I've done this now, with 160 bats in different colonies and different places. And the kinship we can get from genetic markers, and we often have maternal pedigree data. And this is what these fasting trials look like. We pull a bat out, we put it back in so it's hungry. That's just for cuteness, the yawning. The bat's hungry. What's interesting is that the donors often initiate these donations. So it's initiated by the donor. The donor comes over and they get this ritualized belly-to-belly stance, and then the donor leaves their mouth open and the recipient licks the blood out of the other bat's mouth. One thing that's interesting, and this is an example of how I don't understand their sociality, is that bats seem to prefer to be fed by certain partners. So you'll see like offers from one bat, and then they'll be rejected, and then this bat will try to get blood from another bat, and then if it fails, then it'll go back to that bat and say, okay, you can feed me. Whereas other bats have a really hard time getting fed, and they're like begging the females, and they go over to the males, which is what you do when you're really desperate, and they're just, it's really variable. But so yeah, it doesn't make sense that you get these what looks like rejections of donations. But I've only seen this in cases where we have a lot of food sharing. Mostly we're doing these things with strangers where food sharing is very low, so I haven't been able to study that more. We can also manipulate the food sharing, so we can do all these variations on this fasting trial and put a bat inside of a cage and say, if you want to feed this bat, you got to come down to the ground, you got to go in the light, and they'll still do this. So they'll come down and they'll feed them across cage bars, even in situations that they don't want to be in. We don't find any evidence that females groom or share food up or down a dominance hierarchy like you see in many primates. So Rachel Crisp, who was a master's student who worked with me, and Lauren Brandt, she measured over 1,000 observations of conflicts over food. She mapped out the female dominance hierarchy. Females, by the way, are larger than males. They seem to be socially dominant over males, but they don't seem to have that clear of a dominance hierarchy amongst each other. And so this is a comparison of dominant structure in vampire bats compared to other female groups, and you can see vampire bats are last here. Which makes sense because of the up or down the dominance hierarchy, because the food sharing is so reciprocal. So this is a food sharing network from 4 years of data over 1,300 observations. The red nodes are males, the red nodes are females, the blue nodes are males. The size of the node shows how many recipients a bat fed, and the width of the arrow shows the donation rate from one bat to another. So you can see most of the food sharing relationships are among females, which is what you see in the field. This is in captivity where everybody's stuck together with equal opportunity to interact and associate. And under these conditions, and again, the kinship in this group is exactly what you expect in the wild. I don't know if I said that, but under these conditions, the best predictor of how much bat A feeds B is how much B feeds A. These plots are really overwhelming, so I try to think about it from just one bat standpoint. So I've sort of recreated what a typical female's donor network might look like. So if she has a son or daughter, she feeds that pup with regurgitated blood. Sons disperse when they become adults, but females stay, and then it just turns into this long-term mothers feeding daughters into adulthood. Daughters start feeding mothers really early. Even when they're still taking milk and blood, they will regurgitate blood to their mothers as young as 4 months. Then you get these symmetrical food-sharing relationships that occur in other kinds of kin, and then in non-kin, and then you see these weak food-sharing relationships with non-kin. And it's hard to know if they're symmetrical because it's hard to measure symmetry when you have a weak relationship. But I was always sort of puzzled by these because you might wonder if it's true the bats are doing, if they're being nepotistic and they're being reciprocal, why don't you just reciprocate with your matriline, with your family? So why do the bats form these weak non-kin bonds when they already have kin bonds? Why not just reciprocate with kin? So one related question here is there some sort of optimal strategy in terms of having a wider network, we have higher quantity of partners versus maybe a more focused network where you have higher quality of partnerships but not as many of them. It does seem to me from just descriptively looking at the data that bats seem to vary along this continuum. It's definitely not a normal distribution. It's just bats are sort of splayed out on this scale. So one idea I had was, which I called the social bet hedging hypothesis. It's just the idea that the bats are feeding non-kin to diversify their partner investments beyond their maternal kin network, right? So if the social environment is sufficiently unpredictable, you don't know who's going to be there in your tree on any given night, you might want to have non-kin relationships, even if investing in your kin relationships gives you the highest returns per capita, right? That's the best bet hedging idea. Even if you're reducing the mean, maybe you're also reducing the variance. You're buffering yourself against the possibilities of these individuals not being there. So you don't want to put all your eggs in one basket. You don't want your mom to be your only friend, basically. That's more intuitive. So the evidence for this comes from looking at female success under these different conditions. So in a treatment condition, we take away key donor. So if a bat has a mother or daughter in the group, that's the bat we're taking out, because the mother or daughter is always your main food sharing partner. And then the other condition, we take away a non-donor that's never fed you, or we've never seen them feed you. In this case, we've never seen that in the last 4 years. So how well do they cope with this? Does it depend on how many non-kin they feed? So this is what the predictions with social bet hedging look like. So on the x-axis, I'm showing you non-kin fed, but you got to control for the opportunity to feed non-kin, because some of the bats were born into the study and didn't have as many chances to feed other individuals. And then on the y-axis is food received in the present. And so social bet hedging says that under normal conditions, there's no benefit to having this greater network because you have your family there for you. But then when you take away mom or you take away daughter, suddenly these other relationships become important. And that's what we found. So under normal circumstances, females that helped more non-kin did not receive more food. But when we removed their key donor, females that helped more non-kin did receive more food. And so you can think about that as bats with fewer non-kin bonds suffer more major losses when their kin donor is removed. And bats that invested in more non-kin bonds coped better with the loss of this donor because they had more donors, because they fed more bats, because they've been feeding more non-kin. You can't control the size of your kin network. You can't control how many siblings you have when you're born. So these non-kin bonds appear to provide this cryptic benefit that wouldn't be obvious at first if you weren't doing this manipulation, right? You might suggest that, oh, these are mistakes. So this is consistent with responsiveness or reciprocity. And it suggests that bats are investing in specific individuals, not the whole group. And so if you want to think about this as group augmentation, you can't think about it as the group being larger. It's got to be each bat has its own unique network that it's investing in with different relationships. And like I said, when primary kin donors go missing, these non-kin partners can provide these backup donors. But there's a lot here that I kind of just assumed. We don't know. There's a bunch of stuff we don't know. We don't know to the extent to which there's actually a trade-off in are there actually strategies where some individuals are trying to get more and other individuals are trying to be more focused? We don't really know. I mean, I just looked at the data and said, it's consistent with that. We don't know that for bats. We don't know if there's a trade-off across individuals or within an individual. There is some evidence for trade-offs within individuals for quantity versus quality, and that comes from rhesus macaques and lance-tailed manakins. The rhesus macaque evidence was actually in the supplement, so read the supplement when you read papers. That's where I saw that. And there's also evidence that social animals try to replace lost relationships, which is consistent with this idea of them trying to manage their position in their networks. Are animals actively trying to manage the number of relationships that they have, or is it something that's completely not under their control? I think that's an interesting question. So if you want to create more new relationships, how do you do that? How do you create new social bonds? How do the bats form new food sharing relationships? There's this paper, 1998 paper, I don't even remember the name of the cooperative development of cooperative relationships is the name of the paper, because I remember that because my paper was then just in vampire bats at the end. But it's called Raising the Stakes or Testing the Waters. And the idea here is that the way to start a new relationship is you start with small cooperative investments at first to not put yourself too much at risk, and then you gradually increase them in partners that provide high returns. And this is something that humans will naturally do if you put them in an economic style cooperation games, but we don't know if it happens in animals. And one way it might happen is that animals might transition from low cost to higher cost investments, right? So the vampire bats might try clustering with a stranger first to see if they're even tolerated, and then if that goes well, maybe they groom you a little bit, and if that's reciprocated, which can happen immediately, then they might groom more, and then that might develop into food sharing. So this is what I had naturally thought was what, just from watching bats, I thought this is maybe what they're doing. I'm going to show you evidence now for the second part of this, and I'll address the first one. So I haven't talked much about social grooming, but this is a bat grooming itself. A bat that's grooming itself is several times more likely to be groomed by another bat. So they seem to care about what's going on with these bats. They're not just randomly grooming anyone around them. And they also try to remove the bands that we put on them. And if you mess up their fur, those bats will receive more grooming too. And vampire bats are special among bats in terms of social grooming. They spend about 5% of their time social grooming, whether they're in captivity or in the wild. It's like an order of magnitude more than other bats under identical captive conditions. without ectoparasites. No, none of these bats have ectoparasites, except for the myotis, because that's a wild study. Sorry, too much information. So, what am I okay, so this is coming back to the hypothesis. They're starting as strangers. This is what we think is going to happen. Then they're going to groom in this reciprocal fashion, and then it's going to turn into food sharing. You need strangers, so we went to different populations in Panama and captured bats. We held them together, and separately we did these controlled introductions where we introduced 1 to 1 or 1 to 3, and then eventually we mashed everybody together. And during this whole time, we're doing these fasting trials. We did, in the mixed group condition, we did 532 fasting trials over 300 days. There was bats born into the group, and then we watched how they formed relationships too. Most of the bats by the end were social grooming, but only 15% of them had formed food-sharing bonds by the end of the study. We predicted the following pattern. So time is on the x-axis, y is the probability of A grooming B in like a bin of time. So what we thought is that when we see food sharing emerge, if we look back, we should see the grooming rate in the other direction should be going up, but not so much in pairs where food sharing didn't occur. And that's what we saw. So the grooming dynamics predicted the formation of these new food sharing relationships. We replicated this 2 years later in another colony of bats with more strangers, but in a less amount of time. I should say my PhD student did this. The other thing we expected is that if you create a day 0 for each food sharing relationship, you should see that grooming is ramping up before that, but not so much afterwards, right? You're only ramping up grooming when to get to this food sharing. And that's, again, what we saw. So grooming rates increased up until food sharing began, but not afterwards. And we saw, most crucially, that the new relationships emerged in this reciprocal pattern. So when A fed B for the very first time, that made it way more likely that B would feed A for the very first time. So I guess that's the best evidence I have for reciprocity in vampire bats, in case people are curious. If you ask me if vampire bats engage in reciprocity, it kind of depends on what you mean by reciprocity. If by reciprocity you mean responsiveness to your partner's behavior, then I think it's probably yes. But if by reciprocity you mean something much more narrow, like literal tit for tat in a prisoner's dilemma game or something really strategic, then no, of course not. But that's kind of a straw man version of reciprocity, I think. I think the vampire bats do have this fitness independence. I think they do. There are byproduct returns, as I'll show you. There's so many different ways that the behaviors of one individual feed back on another. It's impossible to say that this food sharing is just about the food sharing you get in return. Another thing that I really want to understand that I don't understand yet is we're seeing all these individual differences, but can we use those to predict the outcomes of relationships? So if we've got a bat that's really cooperative with strangers, and another bat that's really cooperative with strangers, does that mean we put them together and it's like they're definitely going to have a cooperative relationship, or can we not predict relationships from individual traits? I don't know why we haven't done this yet. We just haven't done it yet. But that's another thing I really want to know. Okay, so the other thing is clustering to grooming, right? This earlier stage. This is what we've been looking at more recently. So my PhD student, Imran, who's been in Panama, he has a captive colony of vampire bats, he's creating all these new relationships, and his first field season, his PhD, he addressed this question of, can we just play matchmaker? Can we create enduring social grooming relationships by just temporarily forcing bats together into close proximity? And how long would we have to do that? We don't really know, we're just trying things out. So we took bats again this time we had bats from 3 different sites. And we put them together, we let them freely interact for 6 weeks. Then we forced them together in these small groups in small cages for just 1 week. Then we put them back out again in flight cages where they could freely associate for 9 weeks. And so the prediction is that the bats that already know each other, their grooming rates aren't going to increase. The bats that are new, the new pairs, they're going to increase, but not as much as these pairs that we forced together and forced them to just be in close proximity to each other. And that's exactly what we saw. I will say this is when you have a graduate student and you say, make a graph of your predictions, and then they do that, and then you plot their data, and it's like one of the most satisfying things. Sorry, this didn't intend to go off. Okay, so the other thing that was really shocking was how persistent this was, right? So you expect there to be this bump after that 1 week period, but it does seem like this is something that lasts 2 months later, these preferences are still there. So you can think about this as like the college dorm room effect. You get stuck together for a little bit, and you can form a relationship that can last, well after the period in which you're forced together. We saw this with grooming, but we also saw it with food sharing. So bats that were isolated together with another bat were way more likely to start a food sharing relationship in those those happen faster. The other thing that I just threw in my talk for the last minute, but I'm so excited about is that I think I shouldn't even present this because I'm so not sure. There's low, low confidence in this. I think that vampire bats are vocal learners based on these calls that we've measured. We have 800,000 calls of bats with known kinship and social histories, and now we have them together at different points in time. And we haven't done the longitudinal analysis yet, but I was just plotting this data for this grant I'm writing, and I just got so excited. This is really, I can't really take credit for this. This is work done by Grace Smith-Vidaure and Julia Vertilic, and they've been coming up with methods to measure these calls. But basically, when we had bats from different colonies and we put them together, the vocal similarity of their calls is much higher than bats that are from different colonies, and also close to bats from the same, unrelated individuals from the same colony. Okay, so this is the main question. This is the first chapter of my PhD, and I still don't know the answer to this. Every talk that I've given, I think I say, this is the crucial question, and next year I'm gonna, I will have done this, and we still don't know. So we really need to test partner choice. We're supposed to be doing that right now, but we've got problems of the bats they got sick, we got pregnant bats, we gotta wait things out. We're trying to, well, we still don't know if bats choose more helpful partners, but that's one of the key questions. And we don't know if they switch partners, but both of these experiments are ongoing right now. And I know, for those of you who have seen me talk about vampire bats, I basically end all my talks going, and soon we will know whether they switch partners or if they choose more helpful partners. But, I mean, when we started this project, it was before the pandemic, so I can also blame the global pandemic. It's been a nightmare. We all know that. Okay, evolutionary origins of food sharing. How did this evolve? I'm gonna tell a story. I don't know how it evolved, but I think it's all co-opted from maternal care. Vampire bats have extreme maternal care. They're exceptional among bats in terms of their maternal care. Their gestation period is 6 or 7 months, which is extraordinary for a bat. They nurse their pups for 9 to 10 months. They have this long lifespan where they make really high investments in their offspring, and they can live to be 30 years old. I think it might be one of the longest lived non-hibernating bats. That's a lot of qualifiers, though. I don't know. The point is that they make a lot of investment in their offspring, and they have this, high investment, few offspring strategy. And so I think that the evolutionary origins of food sharing are probably similar to these patterns that we see in modern-day vampire bats, where there's maternal care that turns into extended maternal care, which is, it's easy to see how you get nepotism from kin selection. And then all you need to get responsiveness is to just discriminate between relatives that are equally related to. So if you have 2 sisters and one of them you have a better relationship with than the other, is that even possible? And if that's possible, if you are being biased towards kin that are more helpful to you, then you can start to evaluate the quality of your relationships. You can evaluate the returns you're getting, and then that can lead you to start making investments based more on the returns than based on the indirect fitness benefits. And once you have reciprocal relationships forming, you might start to try to form new relationships to widen your network. Oh my goodness, I'm going over time, aren't I? How bad am I still okay? Oh, okay. I forgot to set a timer. So I think all these things are involved. I think indirect fitness is important. I think interdependence is there, and I think there's probably partner choice, although we haven't yet measured it yet. So one of the things I want to do that I'm really interested in is try to understand more about the scale of competition of these bats, because so much of what I've been doing is just focusing on one colony. But we've realized from these studies on social foraging that these bats are interacting with these bats from these other colonies. And to what extent are they competing with bats within their colony versus bats that are strangers in other colonies? And that's something we don't know yet. So this is what we have done already with mapping the roosting networks to the foraging networks with these proximity sensors. And then we started building the cows into the network. So we're making these bat-cow networks. We can put tracking devices on the cows. It's so cool. We can move the cows around and then see how the bats respond to that. And it's just, oh, I love it so much. It turns out it's actually way harder to wrangle cows than I thought. And I thought it would be easy. I thought, well, we got the bat part down, so adding the cows will be easy. But the cows destroyed our equipment. They stomped on our thousands of dollars of equipment, and we had to hire cowboys eventually, and we couldn't actually. The cows have been more difficult than the bats. But they're doing the social foraging. We want to understand how are they regulating this all with calls, and what kind of calls are they using. And this also has implications for pathogen transmission, right? Because we can actually map out all the contacts between bats and cows, and also even other bats that they might meet. The other reason we want to do this is that the other big thing I don't understand is I don't understand the vampire bat's umwelt. I don't understand how they perceive their social environment, right? So they're in these dark roosts, and what is it like for them? Is it like having a dim flashlight where they're like shining it on one bat, they're echolocating it one bat at a time, and it takes a while, and they're like, okay, I know who you are, and I know who you are, and I have to get close and sniff you? Or on the other end of the spectrum, do they have this like super duper social awareness where it's like omnidirectional? I hear all echolocation from all bats. I immediately recognize everyone, and I know that these two are sharing food over there, and those ones are grooming over there. Where along that spectrum are they? I really don't know. I don't know what information they have in terms of how they know where to go and who to groom and where to move on all these different scales. So I want to use these like network microphones and playbacks, and I won't get into it. This is like my grant proposal. I'm so excited. This is like what I'm most excited about, even though I have nothing to say about it. It's just an idea. Okay, that's the end. So take-home message. Oh no, it's not the end. I've got my little conclusions. What factors stabilize food sharing? I think it's all of these things, and I think the question is, what's the relative importance of them, and do they interact, and how do they interact? And somebody help me figure out what that would look like. More theory on interactions, please. That's what we need. How do they form new relationships? I think they test the waters with these escalating investments. And how well do they perceive their social environments? That's, I think, what we need to really tackle next. Okay, so this is the real final slide. I'll end with this reflection about conscious ignorance, how it compels us to look deeper. And I think about that study with Gerald Wilkinson and how he could have just stopped when he saw that 95% of the food sharing is between kin. He could have just written a paper, Kin Selection Explains Food Sharing in Vampire Bats. And none of us would think there's anything more to this system. And he didn't stop there with that simple explanation. He kept looking and testing other ideas. And I think, we have to strike this balance between using vampire bats or your study system just to confirm these sort of theoretical predictions, but also we have to balance that against trying to understand the ecology and describe the full complexity of the choices they face in nature and how they actually make them, and really just do that exploratory work of understanding all of the things that are going on in their worlds that don't have to do with what we're reading about in theory papers. And that requires these really incremental advances, a lot of exploratory work, and really treating science as a marathon and not a sprint. And I will thank my funders, and I want to thank my academic network, and any and all of success that I've had is due to the quantity and the quality of my scientific social relationships. And I hope to grow my network a bit here at this conference, and I thank Animal Behavior Society so much for giving me this opportunity to do that. Thank you so much. I didn't go over time.
Can you hear me? Oh, better. Okay. Thank you, Jerry. That was amazing. We have time for some questions. We would just ask that if you do ask a question, ask it into either this mic or that one. And Gloriana.
Much easier. Thank you, Jerry. Wonderful talk. I was wondering if you had any ideas or predict something about the level of disturbance that colonies could be facing and whether that could influence the number of food sharing partners that they could have. For example, colonies that face very low disturbance, they know that they can constantly rely on the same individuals and therefore just share food with a few of them. Whereas if they're constantly losing some of their partners, they will gain from having more partners.
Yeah, so, I mean, it's an empirical question. I don't know the answer. But, I mean, that's sort of the idea of social bet hedging, is that as your social environment becomes more unpredictable, that you'll try to have a wider network. But don't know if that's true. It's there for somebody to test if somebody wants to do it. And join my lab and work on that if you want. But I don't know the answer. I would love to find out the answer.
Along a similar line, I'm very interested in the social bet hedging, and I'm sorry if this is in the 2017 Biological Letters paper that you mentioned, but do you think with the tracking data that you have, you could see if bats who just consistently roost with the same bats versus those that are constantly bouncing around, if you could see whether they have more or larger social networks with unrelated bats?
The bats that are
I guess
moving less.
bats whose roost mates are less predictable simply because there are more roosts in the area, are they more likely to have kind of like a wider social network?
I think is that what we're working on right now, Liz? I think Liz has an undergrad that's analyzing that exact thing. She has this measure of social unpredictability and trying to use that as a predictor. But I mean, the one thing I'm not sure the answer to that question. I think it's difficult when you think about connectedness because I think being connected in terms of association and being connected in terms of grooming, even though these things are correlated, they're very different things. I have to think about that more. I have to unpack that a bit more, but I don't know the answer to that question.
Great, thank you.
There's a question.
Yeah, it was a really fantastic talk. Thank you. I'm curious about the fact that males don't do this as frequently, and given the fact that losing weight is such a critical thing to survival, are males better foragers, or do they just die?
Yeah,
Because
I am
nobody
so-
feeds them?
I am so glad you brought this up because it's a thing that I thought I should talk about this in my talk, but then I'm like, no, I'll be going off on some tangent about this. This is part of the story that doesn't make sense to me. So, if it's true that this is about survival, you're right. Males should be dying all the time because they don't have these food sharing networks as much as females. And when I talked with Jerry Wilkinson about this, he was like, yeah, the males do die all the time. Like, I think they're dying way more than the females. But then I found this data set in Argentina where they had this really long-term data. And I didn't see any evidence of the males dying off. So then I was thinking, that doesn't make sense. I mean, so this is a mystery to me about what's going on with the males. It could be that males are doing food sharing and we're not seeing it because we're not paying attention to them. And when we're studying these female groups, the males are basically fighting with each other over access to these females. But males also form these colonies that people call bachelor colonies, where it's like a group of males. And nobody really like studies those very much. So we don't know what's going on with the males. And the other possibility is that it's not actually that much about survival. Maybe it's more about reproduction, and maybe it's more about the huge reproductive investments that these females are making, and that puts them more at risk. And so they need this food sharing to sort of bolstering that. But I think that's a really great question that that's an inconsistency with the original story. As I continue to tell the story this way because that's the story that as far as I know, but it doesn't make sense to me that the males are. But the other possibility is that in Argentina, maybe it's different in Argentina than in Costa Rica. I also think we tend to generalize. We study animals in one place and we go, that's what this species does. But just from seeing vampire bats in different countries, I've gone places like in Trinidad, I didn't see any food sharing when I was there. So I don't know if that's just the sample I took or if there's variation in food sharing in different locations. Sorry, I just answered that with like a whole bunch of other things, but it's a great point that doesn't make sense.
You had shown a slide to follow up with that about how long they spend outside the nest. Did you find that the colored bars towards the bottom were the men spending more time outside the nest because they needed to grab blood for themselves more often than the females who may have spent shorter time outside?
Those were all females.
Oh,
We've
okay.
just been ignoring the males, to be honest, because they don't do as much food sharing. And also, very early in my PhD, I fasted a male when I put him back. He got in a fight with another male. He died, and so I thought maybe I shouldn't do that. I shouldn't take away the males and put them back because they do have these territories, and I'm disrupting something else.
Hi, I had a question about senescence, thinking about, it's a matrilineal society, and wondering if the females reproductively senesce earlier, if they maybe have social information that's really useful for the group about foraging, perhaps. Basically, if that could be part of what's driving these relationships.
What was the question? If the females senesce earlier?
I guess I'm essentially thinking about the grandmother hypothesis and other systems where, in elephants and in orcas, where there's sort of a trade-off between being an older female and having social information. So could that explain some of the differences between female networks and male networks and preferential networks? But that would depend on how, I guess, reproductive senescence is happening relative to.
Yeah, I mean, all I can say about that is Jerry Wilkinson got very interested in this grandmother hypothesis, with all the cool stuff with killer whales. And so he dug up all of this data with some greater spear-nosed bats, and I sent him. We didn't find anything we didn't find any evidence for that. But I think we're going to have way more data with age in bats because now we have this epigenetic clock for aging bats. So maybe there's something there, but we haven't found any evidence for that.
Hi. I was wondering, since the mother and daughter have such a strong bond, if the mother started food sharing with a non-kin member, would the daughter also start food sharing with that non-kin member? Would that like increase the chances?
Yeah. So this question of like, is there social inheritance of network ties? I looked at this when that paper first came out. I love that paper. When that paper first came out, I looked at it in my PhD data. But I haven't looked at it again now that we have like way more data. So that's another thing that we just need to investigate. I wouldn't be surprised if that's going on. We did have this one interesting anecdote where a female formed a relationship with another female, became really strong, and they were like very top grooming partners. The first female had a pup, and then she got sick and she died. And then the second female adopted that pup and took care of it and basically raised it until the end of the study. And then we looked back in the literature, and there's this German lab that had a lab of vampire bats, and they had also seen this happen. They saw it happen so many times that they thought that vampire bats were cooperative breeders. And so that's consistent with that idea. But we haven't done the real analysis of just really testing social inheritance of network ties yet.
Great talk, and I apologize if you said something about this and I missed it, but how good is their kin recognition, and do you know like by what mechanism they do? Is it familiarity or scent or what?
Yeah, we don't know. The calls have kinship information in them, which is, as far as we know, is independent of that's what I actually showed you. So bats that are related to each other have more similar calls. We don't know if that's because it's confounded with their familiarity with each other as well. But that's an open question of like the mechanisms of kin discrimination. We don't know, but I'm fairly convinced that they can at least identify their maternal kin. Whether they can identify unfamiliar kin. Quick story: I thought that we had that nailed out, but it turns out we actually put a preprint up, and then we collected more molecular data to strengthen the evidence, and then it just went in the other direction, and we pulled the paper. So if you've seen that preprint, we tried to retract it, but we thought we had evidence for unfamiliar kin discrimination in vampire bats. But then I said, let's just collect more data to make this even stronger because it's kind of borderline. And then when we did, it was like, this is less good. We're like, never mind. So I don't know if they can discriminate unfamiliar kin.
So I'm going to stop the questions there. It's 6:30 exactly. And before we thank Jerry, just one quick announcement. At 6:30, there's an hour free, and then 7:30 to 9:30, we have an awesome poster session, so please join us for that. There will be yummy snacks, aperitivos, lots of awesome science, so please join us for that. And if you have any further questions for Jerry, you can ask him then. So please join me in thanking Jerry for that awesome plenary.
Thank you.